This is part 2 of the lecture notes from chapter 3. So the first type of passive transport process is diffusion, simply diffusion. And when we're talking about cells and the plasma membrane, diffusion across the plasma membrane means that the substance moves directly through the phospholipids, the phospholipid bilayer, rather than traveling through a channel or a carrier protein. So when we say diffusion through the plasma membrane, it's through the phospholipid bilayer. So basically, diffusion is a passive process, so it means that molecules move from their high concentration to their low concentration. That difference between concentrations is called the concentration gradient. So like say, for example, on the outside of the cell, the concentration of oxygen was 10%, and on the inside of the cell, it was 5%. You know, that's a concentration gradient. So also you could call it a concentration difference. So it just means that outside and inside the cell, there's a difference in concentration for a certain substance. So when we talk about moving from high to low concentration, the wording is down the concentration gradient or with the concentration gradient, okay? I'm going to skip that example. So substances that will diffuse directly through the phospholipid bilayer are going to be lipid soluble and small. Molecules that are lipid soluble, such as lipid soluble drugs, alcohol. One of the reasons alcohol is absorbed so quickly. Fatty acids, steroids, dissolved gases, oxygen and carbon dioxide, because they are nonpolar molecules. And nonpolar molecules are typically lipid soluble because lipids are nonpolar. So substances that are going to cross the phospholipid bilayer are either lipid soluble or they're very small, or both. And then ones that are going to pass through channel proteins or carrier proteins. Channel proteins, the substances tend to be very small, water soluble, and ions, which are very, very small substances. They're just a single element with a charge. So water is another one that can pass through a channel protein. And if the molecules are larger and water soluble, they will require a carrier protein. And these will be molecules like glucose and amino acids. So here is a good slide, a good picture that shows you different molecules and how they pass across the plasma membrane. So if they're lipid soluble, like this is a molecule of cholesterol, cholesterol can move through the phospholipid bilayer by diffusion. Channel proteins are used for small molecules that are polar and water soluble. So water and ions like sodium. And then large molecules that are still polar or water soluble can go through a carrier protein, but then there are molecules that can't pass through at all, so they have to be transported by what we call vesicular transport, and that means through a transport vesicle has to carry them across the plasma membrane. Osmosis is a special type of diffusion. It's the diffusion of water. So what's going to happen in osmosis is that water is going to move toward the hypertonic solution. It's going to move toward the most concentrated solution. So let me show you what I mean. Excuse me. The water molecules here in this U-tube in picture 1 are represented by the blue dots, and then there is a solute molecule represented by the purple dots. And you can see that the concentration of solute is lower inside A, and it's higher inside B. So water is going to move toward the higher concentration of solutes. In anatomy and physiology, we say water follows salt because the solute often is sodium chloride, or a solution that contains some type of a salt, S-A-L-T, salt. So water is going to move toward the highest concentration of solute molecules. See how many more of the purple molecules there are inside B? So water is going to move over here, and you see that has happened in picture 2. The water has moved, so the water level is lower inside A now. Excuse me. A solution that is isotonic has an equal solute concentration as what is inside of the cell. And in this case, if you have a cell that's placed into an isotonic solution, the cell is not going to change size. It's not going to shrink. It's not going to swell. It's going to remain the same size. Saline, normal saline is around 0.85%, and that would be isotonic. That's what you see, and I think I've seen it more as 0.89%. NaCl, and that's what you see inside of an IV bag whenever someone has to get fluids, in the hospital. That solution needs to have about 0.85% salt in order so that it won't cause your red blood cells to swell or shrink. In a hypotonic solution, a cell will actually gain water because the hypo means low. And so a hypotonic solution has a lower solute concentration than what's inside the cell. So pure water, distilled water, it's 0% solute, is hypotonic to the cytoplasm inside the cell. And what happens to a red blood cell is that it undergoes hemolysis. It swells up and it bursts, possibly, when it's placed in a hypotonic solution. But if that same red blood cell is placed in a hypertonic solution, that means the solution has a hyper means higher solute concentration than what's inside the cell, then the red blood cell will actually lose water and it will shrink, and we call that crenation. If we know that the solution inside the red blood cell is 0.85%, then if you placed it in a real salty solution, which was like 5% salt, the water is going to follow the higher solute concentration. It's going to move out of the cell and cause the cell to shrink. So remember, hemolysis occurs to red blood cells in a hypotonic solution like pure water, and crenation occurs to red blood cells in a hypertonic solution like 5% salt. So hypotonic, low concentration, hyper is higher concentration. And this is a picture of what happens to the red blood cell when placed in the different solutions. In picture B, the red blood cell is placed in a hypotonic solution of pure water. And so the water is going to enter the cell because it's going to follow the higher solute concentration, which is actually inside the cell. The cell is going to swell. We call that hemolysis. And here, the higher solute concentration is outside the cell. So we say that the red blood cell is placed in a hypertonic solution. Excuse me. So the water is going to leave the cell, and that process is called crenation. Okay, number 8. How would a decrease in the concentration of oxygen in the lungs affect the diffusion of oxygen into the blood? So if the concentration of oxygen in the lungs is lower, it's going to diffuse more slowly into the blood. And 10, relative to a surrounding hypertonic solution, the cytosol of a red blood cell is blank. The cytosol would be hypotonic. If the solution outside is hypertonic, then inside the cell, the cytosol is hypotonic. All right, carrier-mediated transport uses carrier proteins. It can be passive or active. We call it co-transport if you are transporting two molecules in the same direction. or counter transport if you're transporting two substances in opposite directions. Carrier-mediated transport can be facilitated diffusion, which uses a carrier protein but no ATP, and molecules move or substances move with their concentration gradient or down their concentration gradient from high to low concentration. Or it can be active transport, which uses a carrier protein, does use energy, and substances move from their low to high concentration. So they move against. So in active transport, the substances move against their concentration gradient. Okay. Facilitated diffusion is shown in this slide here, and it's showing the facilitated diffusion of glucose. Glucose is soluble in water, so it's not going to diffuse through the phospholipid bilayer since it's water soluble and not lipid soluble. Glucose will, and it's larger, so it's going to need a carrier protein. But notice that on the outside here, the concentration of glucose is higher, and on the inside of the cell, the concentration of glucose is lower. So glucose is still going to move with its concentration or down its concentration gradient into the cell, but it's going to move through a carrier protein. No ATP is required, and so that is that's what you're seeing here. And then you're seeing now that the glucose has been delivered into the cell. So it's moved from high to low concentration, no ATP has been required, and it has used a carrier protein. Active transport does require ATP. It moves substances against their concentration gradient from low to high concentration. We see this with ion pumps that transport ions like sodium, potassium, calcium, magnesium, and chloride ions across the plasma membrane. They're called pumps because they are moving these ions against their concentration gradient from where their concentration is low to where it's high. And then we have exchange pumps, and the most common exchange pump, or the best example, is the sodium-potassium exchange pump, which pumps 3 sodium ions out of the cell for every 2 potassium ions into the cell. And both of these ions are being moved against their concentration gradient. So here's the sodium-potassium exchange pump. You can see that the concentration of sodium is low inside the cell and high outside the cell. Sodium is the purple molecules. But the sodium is being moved from low to high through that exchange pump against its concentration gradient, and it is using ATP to do this. So this is an active transport process. And then the exchange part comes in because potassium is also being moved from low to high concentration, but it's being moved inside the cell. And it's 2 potassium ions that are moved into the cell for every 3 sodium ions out. 3 sodium out, 2 potassium in. And what that does, it keeps the concentration of sodium high outside the cell and the concentration of potassium high inside the cell. Helps maintain a certain voltage across the plasma membrane that we'll talk about in for future chapters. All right, large amounts of materials that can't go through the phospholipid bilayer or carrier or channel proteins have to travel through a vesicle by endocytosis or exocytosis. So endocytosis is when the materials move into the cell through a vesicle. And this does require energy, so it's an active process. The 3 types are receptor-mediated endocytosis, pinocytosis, and phagocytosis. In receptor-mediated endocytosis, a molecule called a ligand binds to a receptor, and then it is pulled into the cell through a vesicle. Cholesterol and iron ions enter the cell by this method, by receptor-mediated endocytosis. And here's how it kind of looks. These are the receptors. The ligands are the purple molecules. They bind to the receptors. The plasma membrane pinches inward and forms a vesicle around those molecules. and then they come into the cell that way. That is receptor-mediated endocytosis.